Okay, welcome, last but not least, to the Pediatric Oncology session. I'm Louis Chesler. I'm the head of the Pediatric Oncology Experimental Medicine Center at the ICR on the Royal Marsden. And we take very much a team science approach and work in a sort of virtual center of excellence very closely with Great Ormond Street, UCL, and other institutions to produce change in treatment of kids with cancer. So I'm going to talk about that approach and about our efforts to bridge that center with resource and expertise here at Oxford through specific funding that's been put in place by the Little Princess Trust, who asked us to do this and asked, why is Oxford, with all of its expertise and its leadership in precision therapeutics and CRISPR that you've heard about over the past 2 days, not more engaged? So this is our effort to do that. And I think we have heard so much about, N=1 therapy for neuromuscular disease and other conditions we would love to emulate in pediatric oncology. The jammy Dodgers that you guys have talked about consistently through this meeting, I think we feel a bit like the crowd sort of peering through the candy store windows looking at the jammy Dodgers, but the door's locked and the windows are a bit foggy. So, potentially we can get in pediatric oncology to N=1 precision therapeutics, ASOs and those kinds of methodologies, but the problem in cancer is a bit more complex to deliver those therapies. So I love that if you come from Oxford, you can get up and say that there's stupidity in an issue and talk about eating dogs, but you can do that because your research is grounded in fact and thought and learning. And so I think that's really respectable, but probably not the approach I'm going to take. What I want to talk about is this. And this is the stupidest problem that remains in pediatric oncology. Ethically, if I could eat a dog to eliminate this problem, I would. These are survival curves for relapsed aggressive brain, solid, and leukemias. And they're all hockey pucks. After 3 years, the survival in many of these conditions that have progressed is less than 15%. And the stupidity of the approach is that 80-some years after we initiated chemotherapy in children at Dana-Farber, we're still taking the same approach of throwing essentially untargeted drugs at this problem to produce no output. And we have very, very little understanding of what the molecular makeup of these tumors is, so we don't really know precisely how to target the biology of these. That produces very little output. And we treat these children when they're rapidly progressive, because in this country there is no molecular screening to speak of for these relapsed conditions. So, what do you expect will happen when you treat a child late with rapidly progressive disease with a drug that isn't mechanistically targeted? They relapse, and that's their outcome, and there's very little you can do to change that. Analogous to the previous session, this is a problem of screening that is easily soluble. Liquid biopsy can measure literally anything in the blood related to the phenotype or biology of cancer. We can do that using a technology invented here, Oxford nanopore sequencing in plasma on a flongle for very low cost. Implementing that in the UK would allow us to rescue probably a third of the children who progress and are lost to disease before we put them on clinical trial. We simply don't have time in that time frame to identify rational therapeutics, and in some cases there just is no availability of rational therapeutics. I'm going to talk about solving these problems. We have no molecular maps of the biology of these. We have very few disease models with which to probe that biology and the development of these tumors. No monitoring, very low access to drugs. So that contributes to loss of children in this scenario. We lose over there are about over 400 relapse cases in this country every year, and that's still the major cause of morbidity in these children. We know that we need unique solutions. We know the biology of these tumors are unique. Most pediatric oncologists are familiar with this. The very low mutational burden in these tumors, very distinct from adult cancer, possibly reflecting the contribution of developmental drivers of disease. So these, identifying those is generally not a problem. We know what they are. Having the resource to target these, that mechanism simply doesn't exist. These are fusions and transcription factors that epigenetically reprogram these tumors to make them progress. Hijacking of normal development. These are developmental disorders, and they act to alter the epigenetic programs of the tissues in which they are generated, and they alter the immune system so that generally these are cold and immuno-unresponsive. So how do we approach drugging these very difficult drivers? I'll talk about one or two cases, ALK and MYC, and where we've successfully managed to do that. What's the evidence that you have these drivers and they actually act as molecular switches that drive the expansion of tumors? This is the data for amplification of C or NMYC in various conditions. So these are embryonal and probably developmentally related tumors that originate in similar tissues of origin, either in peripheral solid tumors or in the brain. And look at the clinical impacts of amplification of those MYC genes. It's enormous. These are by definition high risk across multiple tumor types. Additionally, the data that they corrupt the enhancer networks that epigenetically reprogram these tumors is very definite. So signatures that look like the tissue of origin. This is neural crest. This is a condition called neuroblastoma. These adrenergic transcriptional and enhancer states and mesenchymal states resemble stages of the tissue in which these tumors are generated. So we know we have to target the transcription factors, fusions that drive these programs and make these tumors aggressively progress. And the evidence that not only the epigenetics of these tumors are reprogrammed, but their immunity is also very strong. So a gene like MYC or NMYC, for instance, modulates many of the cell surface proteins that these tumors express, checkpoints, and expression of therapeutically targetable antigens like GD2 or B7H3. So probably the strongest evidence is clinical. This is a patient with neuroblastoma. This is disseminated subcutaneous developing nerve tissue. This represents a developmental aberration. And this is a patient I saw at UCSF early in my career. These patients are called 4S, special neuroblastoma. They developmentally regress. They don't require therapy in large part, and they're almost never NMYC amplified. I think that's strong biologic evidence that the drivers we're looking to drug are molecular switches. So really what we want to do with drugs is flip these molecular switches, induce a restoration of differentiation. And theoretically, if we do that with a targeted drug, we could achieve a durable and even curative outcome. Secondly, conceptually, if these tumors are dependent on continued expression of these driver proteins and they are addicted, epigenetically reprogrammed, and they do not evolve widespread genomic alterations, which is true of many of them, theoretically, one of these precision medicine targeted drugs could give you a durable effect. And we've seen that that's true before. It's true in pediatric oncology with PML-RARA fusions. Retinoids induce restoration of differentiation and increased survival. There are very few examples where we've had access to targeted drugs from pharma that have done this, but it's true of the NTRK fused and dependent cancers. To a certain extent, it's true of the ALK inhibitors. I'll tell you a little bit about those, and some other fusion proteins in other pediatric cancers. So this is where we will act to connect efforts that are ongoing to infrastructure at Oxford that could, I think, inform these efforts. We're going to map the progression of these tumors and provide biomarkers for early screening. This is through a program called SMPEDS, which has been running for the past 5 years, extensively sequences all children who relapse in the country and provided genomic sequencing for children before the NHS and Genomics England did that, and also in the devolved nations. We're going to connect that large data source to the impressive capability of Oxford to analyze data, database it, and present and visualize it. We're going to build preclinical models and connect an existing effort in pediatric oncology where we've built many such models to the Oxford ecosystem, and we'll go through how we will implement molecularly targeted new technology drugs. So this is SMPEDS. This is one of the largest pediatric studies in the country, has run for 5 years, and exceeded its anticipated recruitment, almost doubled it. That was because we delivered information returned to clinicians who could make therapy decisions based on the results of the sequencing. And again, now NHS England and GEL do that so well, but this initiated before that was in place. And many of the tests developed in that program are now implemented in the NHS. So rapid turnaround, high-depth sequencing panels are starting to produce molecular maps of relapse. Our first action in SMPEDS 2, which opens in November and will report changes in liquid biopsy to clinicians in a weekly tumor board, will be to inform them in very great detail about the changes that occur in blood using Oxford Nanopore technology in rapid turnaround. We hope those techniques will be implemented into NHS to make a difference for early screening of kids who relapse. That program is going to involve a high degree of spatial sequencing and immune sequencing, and the integration of that data is a significant challenge. So that we will pair with capability that's present here. This is the existing molecular platform that widely shares the large database generated in these children and allows clinicians to visualize in tumor board molecular changes in the tumors and assign therapy. So this has generated a lot of interesting hypotheses. When you make this data available to people who want to use it, this is a learning system, an AI system, which looking at cell shape changes is capable of inferring the molecular alterations present in these tumors and also starting to make prognostic predictions. This is Oxford Nanopore technology done in the blood. We have several techniques that work in blood at high turnaround. Again, I said in November we're going to start reporting multiple genomic alterations in the blood, including fragmentomics and epigenetic signatures. And one initial finding from this is that detected in the blood, many of our patients, when they progress or relapse, express potentially immunogenic fusions, which if you use the Moderna and BioNTech algorithms are predicted to be MHC associated. So one intriguing possibility from this is that we could initiate patient-specific mRNA vaccination of these children on a short timeline when they relapse. That's an aspirational goal. And that's something that I think with the creative atmosphere that you have here and the capability to actually, in basic and applied immunology, we could approach. This requires a team effort, faster together. This is our large team that executes the sequencing, and we've initiated the connection of the database to the big data center at ICR through Andy Blake and Oxford Cancer Center. So what about the other drivers? And what is the role of mouse modeling in understanding the biology of these tumors? I'll talk about that a little bit. And essentially cut to the chase, which is that we have spent a lot of time constructing molecularly switchable models of these diseases. They're assembled in a mouse hospital. We can administer serial therapeutics to them and watch them evolve. This is a critical platform for the development of drugs. And we've moved, I don't have to tell you about, I think, the impact of immunotherapy. John will talk about that next. It's critical to have mouse models that allow us to test CAR T and immune therapeutics, and that's what these models do. We have strategies in place to knock in human antigens that are targets for therapeutic CAR T. That will be a major step forward. We've placed many of those models at Harwell in Oxford, again, together with a large team that is characterizing these mice in detail using multiple platforms, single cell and spatial technology. That data set will allow these mice to be positioned with great precision next to their human counterparts, and that will become a community resource where preclinical trials can be run. This is neuroblastoma, where the models have been essential in the development of drugs that target two of the major drivers, ALK and NMEK. I'll just go through this very, very briefly. But we were able to construct and understand the developmental etiology of these mice, define an interaction between ALK, which primes NMEK expression, drug ALK, and then drug MYC to a certain extent. That's been successful. Successful inhibition of ALK in the clinic is probably one of the few instances where we've had access to a drug from large pharma and where we've implemented that clinically. That was because these drugs are developed for lung cancer. Patients with mutations in the ALK kinase domain became resistant to those drugs. There's essentially a whole set of drugs available for us to analyze. So we were able to rapidly model the expression of ALK in neuroblastoma. We started doing this in 2008 when that alteration was discovered. And we were able to serially attest a whole set of ALK inhibitors available from pharma. In 2024, the results of the phase 1/2 study of ALK inhibition in neuroblastoma was published with durable and impressive outcomes, although complicated by therapeutic resistance. So the mouse models were critical in that case to deliver that. ALK inhibitors have become a frontline therapy in other pediatric malignancies, including large cell lymphoma. And this is a young lady that we treated many years ago at the age of 7, who is now an adult with a myofibroblastic cure in which she achieved a complete remission. Currently, a minority of patients respond in this way. Our task is to increase the number of patients for which these drugs are available. So how do we target the other drivers? I'll go through sort of 20 years of trying to do this. It's a very challenging target with a disordered protein structure that is essentially impossible to drug using conventional techniques. Just by saying that we understand the mechanism now by which this oncogene works. It invades enhancers. Present on enhancers that support the transcriptional circuit of neuroblastoma is CDK9, which binds to the Pol2 complex and to MYC. And our first drug synthesized at the ICR that inhibits that target went into clinical trials about 3 years ago, now with a second generation, more potent selective CDK9 inhibitor following that. There are other mechanistic combinations that we have developed and I won't discuss. And what I just wanted to conclude with was a better approach to targeting these drivers. How will we do that? And we're lucky enough to have been awarded a grand challenge in pediatric drug development recently, this year. There were 2 of those awarded. And this is ours. This is an exceptional team of medicinal chemists, structural biologists across the world. The center of this program is the ability to generate fragments against these difficult to target drivers. That will be done at Diamond Light Source on the Harwell campus by Frank van Delft. And the approach there is to blanket these structures with fragments that could serve as the vectors to produce protax degraders and small molecule drugs. And now we have the resource actually to target these difficult proteins. There are more aspects to this program that we're quite proud of. Significantly, there's an advanced program of T-cell engineering, which John Anderson's going to talk about next. And really the central concept of that is that if we develop a glue or degrader drug that we can treat a child with, he is building CAR T cells that are responsive to that glue because the CAR T switch either turns on or turns off. And there's a lot of other resource that comes through that program that, again, will connect us through drug development with Oxford. The announcement that this grand challenge was funded was made recently. This is a novel model for drug development where we're paired with LifeArc, who have a lot of experience in the development of major cancer drugs and have committed 100 million pounds to pediatric oncology drug development. So this is an academic charitable partnership. You'll hear more about other approaches that are being developed in Cleveland to do that. LifeArc put another 5 million pounds into this team to be able to deliver these novel drugs. So we're very proud of that. And then I would just conclude that, with a combination of early screening and precise screening and rapidly adaptive clinical trials that include combinations of these immunotherapeutics plus glue degraders or targeted drugs, we can change the way these relapse trials are done. We can detect patients early, treat their disease before it progresses, potentially eliminate it with an immunologic intervention, and that should increase outcome for children. And I'll just finish here by saying that if you provide a patient with a targeted drug, as well as certainty in a biomarker test that informs them about the progress or remission of their disease, that's incredibly empowering. And some of our patients have told us this is the best thing that the NHS has done. This is a young lady who experienced multiple relapses, responded to ALK inhibition, and is now free of disease for over a year. So we aim to do that more. So the talks that follow will, I think, illustrate aspects of what I've said, which is that there are rapid advances in immunologic therapy and CAR T. John will talk about that. Potentially the ability to intervene, screen early and treat early. Sarah Blagdon from Oxford will talk about that. And novel models of academic partnership with industry and organizations like the Harrington to produce new drugs. I'll stop there. I'm sort of 4 minutes over, so you can take an urgent question if there is one. Otherwise, we can introduce John to talk about CAR T.